Hello, everybody, and welcome back to BioSC 140 Human Physiology. This video will be the experimental procedures video for Laboratory 25/26 Respiratory Physiology. We're going to cover FOX 8.1, FOX 8.2, and the BioPack Lesson 8. You'll find this all in the supplement also. So, spirometer, measuring vital capacity, and respirometer, tracings in supplement. All right, so FOX 8.1, vital capacity. Vital capacity. So first off, what is vital capacity? What is vital capacity? So vital capacity is the amount of air from complete exhale to complete inhale. So exhale as much as you can. And now inhale as much as you can. That is vital capacity. Or with how we're going to do it with these two ways, dry and wet, it's breathe in all the way. So at that point, how much air do you exhale from complete inhalation to maximum exhalation? So vital capacity, max inhale and then blow out through the spirometer. So right here, we have these in lab. It's a dry spirometer. Why is it called dry? There's no water involved. Also, sometimes they're called handheld spirometers, but right here. In lab, we use a different mouthpiece for each person. And the way you use it is you set the dial to 0, you breathe in all the way as much as you can, and then you exhale as much as you can. You don't want to exhale when you use these super fast. It needs to be like a stable, controlled, like medium speed exhalation. And you exhale out as much as you can, and the dial will give you a reading for your vital capacity. With the wet mechanism, the wet spirometer, it's a little bit different. So we actually have this exact one in lab. And the way it works is you exhale into this mouthpiece here. Your air goes through this tube, and then that tube comes up underneath this, what we call bell right here. So it's like an upside down box. This box is open on the bottom, and then, the 4 sides and the top are all, airproof. So you exhale, and the air comes through this tube and up underneath this bell. And as you do that, the bell will rise because the bell is sitting inside this larger chamber that's full of water. So larger chamber full of water. This right here is placed inside of it. You exhale, air goes underneath the bell, and it causes the bell to rise up. As it rises up, you can see that there's this chain here. Well, this chain goes up as well and moves over this pulley, and there's a measuring tape right here, a ruler. So you can actually see, so for every 1 inch that the bell rises, that chain's going to move 1 inch to the left. So you can actually see how many inches up the bell moves as you exhale into this wet spirometer. And then you have to do some math, because just knowing, the bell rose 3 inches doesn't tell you how much volume your vital capacity is. So let's think back to algebra. Let's think back to geometry. How do you get the volume of a cube? Or the volume of a cube. It's length times width times height, right? You get volume with length times width times height. Well, we can measure the length, we can measure the width, and then as we see how far up it goes, we can measure the height. So we get, the 3-dimensional shape or volume of all the amount of air we exhale. Now, the length times width is going to be 1,000. The height is going to be however high the bell rises, so it'll be different for each person. So you multiply how much it rises by 1,000, and then you multiply by the BTPS correction factor. So the volume of a gas changes with temperature. The volume of a gas changes with pressure. There's different factors that come into play when determining the volume of a gas. And so we have to use this correction factor. Mainly it's because inside of our bodies, it's a different temperature than inside the bell, so it changes the volume some. So we multiply, 1,000 by how much it rises by 1.1, and we will get the value of our vital capacity. We're then going to compare, if we were in lab, we would compare our actual calculated vital capacities with our expected vital capacities. Be ready to determine that on the quiz. There are some practice problems with it. So right here is Biopack Lesson 8. And for Biopack Lesson 8, we're going to have two sensors. We're going to have two data collecting devices. We're going to have this chest strap, which measures breathing rate and measures the depth of breathing. So right here on this picture, you can see there's this strap around this person's chest. And what it does is it measures chest excursion. It measures how much your chest expands, like your rib cage expands and contracts through a respiratory cycle. And then we're going to have this other wire right here taped onto our subject's face with a little thermometer right underneath their nose. The thermistor is that thermometer, and it's going to measure airflow temperature. So the thermistor measures airflow temperature. We're going to breathe in cool air. Air is going to pass that thermometer. We're going to see that temperature reading on our graph on the computer. And as we exhale, we're going to exhale warmed up air. Our bodies are warm. The air is inside of our lungs is going to warm up. So we're going to exhale that warmed up air past the thermistor, and it's going to make the temperature rise on our graph. Apnea is normal breathing. We're going to see how the graph looks, how the temperature changes are, and how chest excursion, chest movement is with apnea, normal breathing for 20 seconds. We're then going to have the person hyperventilate. They're going to take rapid, deep breaths for 30 seconds. Actually, we don't do it for 30 seconds. We do it for about 3 or 4 breaths. Safety first. We don't want anyone to pass out. So hyperventilation, rapid, deep breaths. We're then going to have the person do hypoventilation, slow, shallow breaths. And we're going to see how the temperature and chest movement graphs are different in those different situations. How do you think the graph is going to look with apnea for temperature and chest excursion? How do you think it's going to look for hyperventilation? How do you think it's going to look for hypoventilation? So we're going to look at the respiratory cycle for apnea and hyperventilation and hypoventilation. We're going to calculate the duration of inspiration with each one. We're going to calculate the duration of expiration with each one, the total duration, and breathing rate. We're going to look at temperature changes also. So breathing rate, BPM, breaths per minute. Ventilation depth, so we're going to do peak to peak. So peak to peak would be for one cycle. You're going to have, one cycle. Perfect. Some data processing. Peak to peak. We're going to look at breathing rate for apnea, hyperventilation, hypoventilation, and we're going to look at the amount of chest expansion that we have for each one, or ventilation depth. We're also going to look at how the temperature changes. How are the temperature fluctuations with these different situations? How is the temperature graph going to change with apnea, hyperventilation, and hypoventilation? Perfect. And I will see you all in the discussion video. As always, let me know if you have any questions. I am here to help. See you in the next video.